Enough is enough. I have had it with these mother effing snakes on this mother effing plane. I'm sure a lot of you undoubtedly heard that famous line by Samuel L. Jackson in the 2006 film Snakes on a Plane. Probably even with more colorful language than I used here. And perhaps many more are deathly afraid of snakes and all things that slither. Are snakes really all that bad? What if snake venom could be used for good? In the 1960s, scientists learned that venomous snakes didn't have to cause chaos on planes. Their venom could actually be used for good things. And this caused a pretty big medical revolution. So let's go back to the 1960s. Snakebite victims of the Brazilian pit viper, Bothrops jararaca, weren't just in pain. Many were going into a rather dangerous and sometimes fatal drop in blood pressure. Doctors couldn't explain why the crash was so severe and even so fast. So enter Sergio Ferreira. He's a Brazilian pharmacologist who wanted to know what exactly was in this venom that could do that to the human circulatory system. Ferreira found a family of small peptides in the venom that dramatically potentiated bradykinin. And bradykinin is a 9-amino acid peptide that your body already makes to dilute blood vessels, or dilate blood vessels. That means make them larger. He called them bradykinin potentiating peptides. We can abbreviate that BPPs for short. But how were they doing that? To understand it, let's zoom out and discuss the system that regulates blood pressure in your body every second of every day. This system is called the Renin-Angiotensin-Aldosterone System, or RAAS. It's essentially your body's blood pressure thermostat. So first, when blood pressure drops or the kidneys excrete low sodium, specialized cells release an enzyme called renin into the bloodstream. And renin acts on a protein that's made by the liver called angiotensinogen. It slices off a fragment of that called angiotensin I. Now, on its own, angiotensin I is basically inactive. It's a precursor, kind of waiting to be armed. That's where ACE, angiotensin-converting enzyme, comes in, mostly located in the small blood vessels of the lungs. ACE clips off 2 amino acids from angiotensin I, and that converts it into angiotensin II. And this is a critical activation step. Now, angiotensin II is a very powerful enzyme. This is where most of the magic happens to regulate your blood pressure. First, it increases sympathetic nervous system activity by facilitating the chemical release at nerve endings and stopping normal reflex heart rate reduction in response to high blood pressure. Next, it also signals the adrenal glands to release aldosterone. This tells the kidneys to retain sodium and water and to excrete potassium. This raises your blood volume. And it also directly constricts blood vessels, raising the blood pressure almost immediately. And lastly, it triggers thirst and the release of antidiuretic hormone from the pituitary gland. That's the gland right at your brainstem, and that promotes water retention. So put together, the renin-angiotensin-aldosterone system is a feedback loop, and that's built to raise blood pressure and to keep it there. It's essential when you're bleeding out or you're dehydrated. But in people with chronic hypertension, this system is often overreactive, quietly keeping blood pressure elevated year after year. And that puts added strain on the heart, the kidneys, and the blood vessels, and that leads to cardiovascular disease. So, this raises an obvious and interesting question. What if you could block that one conversion step? What if you could turn off angiotensin II production at the source? That question is exactly what the Venom research had already answered. Scientists just didn't know it yet. So do you remember those bradykinin-potentiating peptides that Ferreira isolated? Well, it turned out the enzyme responsible for breaking down bradykinin, for deactivating it, was the same enzyme that was responsible for activating angiotensin II, or ACE, right? This enzyme is teprotide, and it's essentially an ACE inhibitor. So this teprotide found in the Venom was inhibiting ACE directly. If you block ACE, two things happen at once. Number one, bradykinin, a vasodilator, sticks around longer. And number two, angiotensin II, a vasoconstrictor, stops being made. Blood pressure doesn't just drop, it gets hit from both directions. And so that's the aha moment. A single enzyme sitting at the crossroads of two blood pressure pathways and a snake had already evolved a molecule to shut this down. Okay, but we have a little problem here. You can't just inject snake venom peptides into hypertension patients for the rest of their lives. The original peptide, that teprotide, worked, but it only worked intravenously. It didn't work orally. And see, you can't take peptide drugs orally because, first of all, they're going to be destroyed by powerful protease enzymes in the stomach. And secondly, they're just a little bit too large with too high molecular weight to be absorbed by the intestinal epithelium. They have to, in order to be absorbed, you have to break it down into individual amino acids or maybe dipeptides or tripeptides. Very short, very small things. So, a team at Squibb Pharmaceutical Company, David Cushman and Miguel Ondetti, they built on the pharmacology by John Vane. They set out to design a small molecule based on peptide structure that captured the venom peptide's effect in a form that you could swallow. That would be orally. The scientists observed that peptides in snake venom consistently ended in proline. That wasn't just a coincidence. ACE has a strong structural preference for proline sitting in its terminal binding pocket. This is actually why bradykinin and angiotensin I resist the full breakdown by other enzymes. Proline's rigid ring fits very neatly into that pocket, and this anchors the whole molecule in place. But, we're a little earlier, there's no crystal structure of ACE that was available at the time. That wouldn't be available until about 2003. So the team modeled the active site by analogy to a related, well-studied enzyme, Carboxypeptidase A. And this also used a zinc ion to catalyze its reaction. They didn't really make a true homology model since the sequence identities were not close enough, but the active sites were similar enough. They each contained histidine and glutamate residues, so they could bind very similarly. Now, the first attempts at making an inhibitor were built around proline anchored to a succinyl group. They turned out to be rather weak inhibitors. They did make a pretty big breakthrough, though, and it came from separate research showing that a thiol, or a sulfhydryl group, binds zinc far more tightly than the carboxylate group that they'd been using. Swept that pace in, and the potency jumped rather astronomically. Add a methyl group for extra potency, and so now you get the 3D orientation, the stereochemistry exactly right. And you land on the final structure of the drug, Captopril. Captopril was approved by the FDA in 1981 as the first orally active ACE inhibitor in the world. And it's a drug that was designed rationally, piece by piece, around the natural molecule found in snake venom. Captopril didn't just become a successful drug, it actually became a template. And it's the prototypical drug for an entire class of drugs called ACE inhibitors, angiotensin-converting enzyme inhibitors. And so this followed in its footsteps, most swapping out the thiol group for a carboxylate-based zinc binder to reduce side effects like taste disturbances and skin rash that were common in Captopril. And so for many years, ACE inhibitors were among some of the most widely used and prescribed medications on earth, commonly used for hypertension, heart failure, kidney protection, and diabetes. The top-selling drugs in 2025 today, they're mostly GLP-1 inhibitors and anti-obesity drugs, but the global market for ACE inhibitors remains at about $7.41 billion. And that's expected to grow to about $10.1 billion by 2030. So this is one of the clearest examples in modern medicine of structural-based rational drug design. Study a natural toxin, understand the biology it's exploiting, and reverse engineer that mechanism into a life-saving therapy. Not bad for something that started with a snake on a very different kind of flight, from the venom gland to the bloodstream to one of the most important prescriptions in cardiology history. I think even Samuel L. Jackson would probably approve. So I hope you found this video interesting and informative. And if you like the video, go ahead and hit that little like button down below. That's very helpful in monitoring our progress here, monitoring what people think of these videos. If you want to see more content like this, hit the subscribe button on my channel. I think that's down here. And hit the little bell icon, and that kind of notifies you of upcoming content. If you do have any questions about the Renin-Angiotensin-Aldosterone System or drug discovery in general or anything else, go ahead and leave that in the comments below, and I'll see you in another video. So thank you for watching.